Okay, so let's do a sample sequencing problem. So in this problem, I'm going to give you some data, and then I'm going to walk you through how I solve it. But you got to keep in mind that everybody approaches these types of puzzles differently, and there's no right way or wrong way. But I'll try and give you my thought process as a way to sort of model what you might be trying to do. So in this example, I'll tell you that we have a 7-mer. So mer means 7 amino acids. So we have 7 amino acids that we're going to try and sequence. So the first experiment we do is amino acid hydrolysis or amino acid analysis, and we find out the residues that are present. So we have an asp, we have leucine, we have lysine, we have methionine, we have phenylalanine, and we have tyrosine. So I'm telling you, that's the data that we got out of amino acid analysis. So immediately what you should say is that there are 6 amino acids, but I'm telling you it's a 7 amino acid protein, that one of these amino acids is a double. So that's one piece of information that you should pick up right away. Now you might be thinking, well, I thought amino acid analysis should give you relative amounts. And yeah, it should, but not all data is perfect. So keep that in mind too. This is just the data that you have to work with. So the first piece, amino acid analysis, gives you these 6 amino acids are present. The second piece of data that you get is that trypsin, when added to your protein, had no effect. So immediately what that would tell you is you would look in table 3.6 and you would find out, okay, trypsin cleaves on the C-terminal end of lysine and arginine. We know that the only one of those 2 amino acids we have present is lysine. So this tells us immediately that our lysine residue is on the end. So the only way we would get no effect, most likely, is if the K was at the very end of the protein, so there's nothing to cut it off from. So the third piece of information that I'll give you is that after one round, so just one round of Edmond degradation, you identify the following structure. It looks like this. So bear with me while I draw this sucker out. Okay, so again, you can get in a piece of immediate information from this. So what you should be looking for is if this is at one round of Edmond degradation, this is a PTH derivative, so you have to find the amino acid. So here is the amino acid, which tells us this is our side chain. So this tells us immediately that the N-terminal residue, the first residue in our sequence, is phenylalanine. Okay, so now I'm going to give you some more protease experiments. So the fourth piece of information is that if you treat your protein with chymotrypsin and you run the products either over HPLC or maybe you do an SDS page, you get a dipeptide, so that means it has 2 amino acids, and you get a tetrapeptide that has 4 amino acids. Then, if you do amino acid analysis of the tetrapeptide, you find out its composition. And its composition is leucine, lysine, and methionine. So notice that I'm using commas here. So when you see commas between amino acids, that just means they're being listed in terms of composition. It doesn't mean that that's the order. So again, in this example, we know that if this is a tetrapeptide we sequenced, and it only has 3 different types of amino acid, then one of these is the one that is the double in our protein. We also know that lysine here, we already identified that as being on the end because we didn't get any results for the trypsin digest. So that's suggesting that lysine is not the double. So the double is either the leucine or the methionine. So these are just pieces of information that we're getting as we go along. So the last piece of information I'll give you is that treatment with cyanogen bromide, which, technically is not actually a protein, it's a molecule, but it still accomplishes the same thing. Treatment with cyanogen bromide yields a dipeptide, it yields a tetrapeptide, and a free lysine. Okay, I paused this during my office hours, and I'm not sure what I said last. So hopefully what I said last was that in this fifth data set, we did a treatment with cyanogen bromide, and we got this dipeptide, this tetrapeptide, and free lysine. And so I'm not totally sure how I'm going to throw that into my analysis yet. So I'm going to move forward and show you how I would approach now the next part of solving this problem, now that we've extracted what little pieces of information we can from our data sets. So first of all, I've got a 7 amino acid protein. So 1, 2, 3, 4, 5, 6, 7. This is very hangman-like. So we know, first of all, that trypsin had no effect. So we know that there's a K on the end. And I guess I'll add too, I like to keep track of who my amino acids are. So I know I've got an aspartate, I've got a leucine, I've got a lysine, a methionine, phenylalanine, and tyrosine. And one of these is a double, so it's not like I can cross off anything yet, so we know who we've got. So trypsin had no effect, lysine on the end. We know that the N-terminus was phenylalanine, so I'm going to put that in the front. We know then that chymotrypsin, so the chymotrypsin, if you go to table 3-6, it cuts on the C-terminal end of aromatics. So it cuts on the C-terminal end of phenylalanine, tryptophan, which we don't have any, and tyrosine. Okay, so let's take a look then at what this means. And I think I can also go back and cross out lysine as being used because we also figured out that lysine is not our double earlier. So if I put, we know that with the chymotrypsin we're going to get a dipeptide and a tetrapeptide. And we know that we're going to cut after this phenylalanine. So if I come here and I put a cut there, well, that tells me right away I've already got one amino acid floating off. So it just didn't show up in the data that we got a dipeptide, a tetrapeptide, and a single amino acid, but that's okay. So I have also then got this tyrosine. So if we put the tyrosine right here in this third position, and we cut after it with chymotrypsin, that would give me a dipeptide and a tetrapeptide. And we know from our information about chymotrypsin and the composition of the tetrapeptide that it does have lysine, so this way makes sense so far. We also know that it does not have aspartic acid, so that means that the aspartate would have to go here. That also tells us then that these last three positions have to be a combination of leucine and methionine. So now we know that leucine or methionine has to be our double. So then we go back to our cyanogen bromide data and we look at table 3-6 and we see that cyanogen bromide cuts on the C-terminal end of methionine, so it cuts after methionine. And we know that when we do this cyanogen bromide, we get a free lysine, so that's suggesting we're going to, if we put the methionine there, and we know that if we cut after it, we'll get a free lysine released. But we also know that we get a dipeptide and a tetrapeptide. So that means our methionine has got to be our double because we've got 1, 2, 3, 4, 6 amino acids that we've got to break apart into a dipeptide and a tetrapeptide. So continuing on with our yellow cuts, we can't cut here to get a dipeptide and a tetrapeptide because there's an aspartic acid there. So let's try putting our methionine then in the other possible location, which is here. And then we would cut at this position. And so this would be our tetrapeptide, this would be our dipeptide, and this would be our free lysine. And then that leaves us leucine, in the last position. So the way you solve these is sort of by trial and error. So you go back then once you've got the data. So I have a potential sequence here for my small peptide. Then you want to go back and do the cuts again and make sure that the sequence you have is in line with your data, that it makes sense. So that's solving a basic sequencing problem where you don't have to worry about location of disulfide bonds. So I'll do that example in the next video.